We've all heard a million times that DNA is the blueprint of life. But if you really think about it, a blueprint doesn't just magically sprout into a fully formed building all by itself, right? It needs materials, it needs an environment, it needs construction. Well, in this explainer, we're going to completely demystify the invisible rules of genetic inheritance. We're going on a journey straight from your basic genetic blueprint all the way up to how we can actually predict the wildly complex outcomes of life. So, let's jump right in.
Okay, so here's our roadmap for today. First, we'll cover the phenotype formula. Then, how genes interact. Third, the over-dominance advantage. Fourth is epistasis, masking the genes. Fifth, predicting genetic probabilities. And finally, number 6, decoding genetic crosses.
All right, let's kick things off with Section 1, the phenotype formula, the master equation of life.
To really get genetics, we have to establish the absolute foundation of what makes any organism function. Your phenotype, which is just a fancy word for the final observable physical form of an organism, is the result of one very specific formula. Genotype plus environment plus the interaction between the two.
Let's break those variables down a bit more because it's absolutely crucial. G stands for genotype. That is your internal, hard-coded genetic blueprint. E stands for environment, meaning all the external factors out there, like the amount of sunlight a plant receives or the daily nutrition an animal gets. And then we have G times E, which represents the complex interaction between the two. The main takeaway here is that your genes don't just operate in a vacuum. They act way more like building materials that are constantly reacting to the environmental weather around them. You literally cannot understand the final living organism without understanding both the blueprint and the environment it was built in. And yes, this goes for plants, animals, and for sure, even us humans.
Moving right along to Section 2, how genes interact, the complex web.
So, now that we've got the macro-level formula down, let's zoom in a little bit. Our biological instructions communicate in a whole multitude of ways. First, we've got linear interactions, where genes are just interacting on the exact same chromosome. Then there are allelic interactions, which is where a gene interacts with its corresponding partner in a homologous chromosome. Next up, epistatic interactions. This involves a gene in a heterologous or entirely different chromosome altogether. And here's the wild part. Genes don't just talk to other genes. We also have internal interactions with biological factors like hormones and external interactions with outside forces like light and nutrition, which, you'll notice, pulls us right back into that big phenotype formula we literally just covered.
Let's actually zero in on that allelic interaction for a second. Specifically, what happens when there is a total lack of dominance? Picture this. If a certain animal has a double capital L genotype, it gets long ears. A lowercase double L gives it short ears. But if you combine them into a mixed capital L lowercase l genotype, you'd probably guess it gets medium ears, right? Actually, no. You get a completely earless bunny. It's such a perfect, fascinating showcase of how gene combinations can create entirely unexpected results when neither gene steps up to be the dominant boss.
That brings us to Section 3, the over-dominance advantage, survival of the heterozygote.
Now we're going to look at a super high-stakes scenario where being a hybrid actually gives you a massive biological edge. This concept is called over-dominance, or sometimes you'll hear it called heterosis or hybrid vigor. Have you ever wondered why mixed-breed dogs are often way healthier than purebreds? Well, this is the exact genetic reason why. A heterozygote, which just means an organism carrying two completely different alleles for a trait, actually possesses greater biological resilience and vigor than an organism stuck with identical alleles. It sets up this really profound idea that sometimes the toughest, most robust organisms out there are the ones carrying a mixed bag of genetic instructions. To really see this high-stakes reality in action, let's consider sickle cell anemia. If you have a double capital S genotype, you get normal red blood cells, but unfortunately, it leaves you highly vulnerable to malaria. Now the lowercase double S genotype gives you sickle-shaped cells, causing anemia, and guess what? It still leaves you vulnerable to malaria. But what happens with a mixed capital S lowercase s genotype? Because of heterosis, this specific mixed combination actually creates a biological resistance to malaria. It brilliantly proves why mixing generic instructions can create an absolute survival advantage when you're placed in the right environment.
All right, Section 4, epistasis, masking the genes.
We are shifting gears entirely now to a completely different type of interaction. This is one where one set of instructions just ruthlessly overrides another. We call this epistatic interaction. You can basically think of it as the ultimate genetic veto power. It happens when a gene or maybe a pair of genes in one chromosome steps up and completely alters or masks the expression of a gene in an entirely different chromosome.
The original instructions? Oh, they are still sitting right there in the blueprint, but they've been totally silenced.
To see this veto power in full force, let's look at horse coat colors. A horse might have the underlying base genes to be beautifully black or chestnut, but anytime an epistatic gene for white, represented by that capital W, is introduced into the mix, it instantly masks the base coat. It literally doesn't matter what the base color was trying to do anymore, the epistatic white gene has completely vetoed it, turning the final horse completely 100% white.
Section 5. Predicting genetic probabilities, the mathematical tools.
Okay, so far we've mostly been observing the rules of genetics. Now, let's actually calculate them, using one of the most famous tools in all of biology, Reginald Punnett's classic square. By simply placing the dominant and recessive alleles of one parent along the top and the other parent along the side, this brilliantly simple grid perfectly maps out the probability of offspring inheriting those heterozygous or homozygous combinations we've been talking about.
But as you can imagine, when we start dealing with multiple traits all at once, a Punnett square gets a little bit unwieldy. That's when we have to apply multiplication and addition rules using a much more advanced tool, the forked line method. In something like a tri-hybrid cross, this method allows scientists to mathematically map out the diverging probabilities of 3 entirely different traits combining all at the exact same time, which results in some highly specific genetic fractions. It's like a giant biological puzzle coming together.
And finally, Section 6. Decoding genetic crosses.
So now that we have the math down, let's look at the specific breeding experiments, which we call crosses, that scientists actually use to uncover these hidden genotypes in the real world. A monohybrid cross is the simplest one. It focuses purely on the inheritance of one single pair of alleles, like just tracking whether a coat color is going to be black or red. But as we scale our predictive models up, we introduce the dihybrid cross. This tracks 2 completely different traits simultaneously. For example, figuring out not just the animal's coat color, but also whether it's going to be horned or polled.
Then we have something called a backcross. Now, this is a very specific strategic move where a hybrid offspring is bred back with one of its original parents or an animal that is genetically identical to that parent. Why do they do it? It's heavily used to reinforce specific, desirable parental traits right back into a hybrid lineage. And last but not least, the test cross. Think of this as the ultimate genetic lie detector test. Imagine you're looking at an animal showing a dominant trait, but you have no clue if its internal blueprint is purebred or if it's secretly carrying a recessive gene. Well, by crossing it with a strictly homozygous recessive animal, the resulting offspring will instantly reveal the genetic truth hiding in that blueprint.
Wow, what a ride. We've journeyed from the foundational equation of phenotype, we've explored the incredible survival advantages of overdominance, and we've uncovered how epistasis can completely mask hidden codes. So before we wrap up this explainer, I want to leave you with a provocative thought. Based on all the principles we just broke down today, what invisible genetic interactions do you think are masking traits inside your own blueprint right now? Thanks so much for exploring the amazing, complex world inside your genes with me today.